The fire had been burning for 2 days without going out. She kept it alive the way her mother had, the way her mother's mother had, banking the coals under a layer of ash each night so that in the morning a single breath would bring the orange back. She was not thinking about the fire as something precious, something that required thought. It was simply one of the things she did, the way she breathed, the way she scanned the edge of the tree line without moving her head. Her name, if you could call it that, was a sound made at the back of the throat, a sound that meant something specific to the 7 other people who lived in her small group, and nothing at all to anyone else. She was 31 years old, which meant she was one of the oldest people in her group, and she knew things. She knew where the best ochre deposits were. She knew which routes were safe after the rains and which would make the stomach cramp. She knew the paths of the animals through the dry season, and she could pass none of that knowledge on, not completely, not with the precision that would have mattered. She could gesture. She could demonstrate. She could produce a small set of calls and signals that carried emotional content, urgency, direction. But she could not say, the routes on the western slope near the 3 flat stones are safe in the wet season, but not in the dry season, because the soil chemistry changes and the toxins concentrate in the tuber. That sentence, or anything like it, was beyond her. Not because she was not intelligent. She was. It was because something inside her skull, something in the architecture of her throat and in the wiring between her mouth and the part of her brain that planned sequences of movement, was not yet arranged the way it needed to be. Somewhere in that same body, in every cell of it, there was a gene, a stretch of DNA that, in her version, looked very much like the version carried by the chimpanzees that occasionally screamed from the fig trees to the east. That gene would be named FOXP2 by scientists who would not exist for another 800,000 years. In her, it produced a protein that shaped the development of certain neural circuits. Those circuits controlled, among other things, the fine motor sequencing required to produce complex, rapid, voluntary sounds with the lips, tongue, soft palate and larynx working in precise coordination. In her, those circuits were functional but limited. They produced enough control for the calls and signals she used, not enough for language. 250 kilometers to the northeast, there was a man who was her distant cousin, though neither of them knew the other existed. He was 24. His group had been following a herd of cattle-like animals for 6 days. In the back of his throat, in the nucleotides that made up his version of that same stretch of DNA, there was a single position where the chemical base was different. Not dramatically different. 1 letter in a sequence of billions. Cytosine where there should have been thymine, or something functionally equivalent in the language of molecular biology. That single substitution meant that the protein his version of FOXP2 produced was slightly different from hers. And that slightly different protein, during the years his brain was developing inside his skull, had wired certain circuits in a slightly different way. He could not speak a human language either. Not yet. That change was not the whole story. It was a first step in a process that would take tens of thousands of years to express itself fully. But the mutation was there. It existed now in the world, in one body, in one man following a herd across a dry plain. And it would not disappear. This is a documentary about that mutation, about what it changed, about what it set in motion, and about the long, slow, uneven process by which a single variation in a single gene contributed to making the human animal into something that no other animal has ever been. The channel you're listening to is dedicated to real facts about our Earth, past civilizations, and the deep history of the human species. This documentary was made for listening to while you fall asleep or settle into rest. The pace is calm and steady. There is no urgency here. You can let your attention drift and come back and drift again, and the story will still be there, still moving at the same unhurried pace. The topic tonight is one of the most consequential events in the biological history of our species, a mutation in a gene called FOXP2, and the long chain of consequences that followed it. Everything you will hear is based on real genetic, archaeological, and paleoanthropological research. Where the evidence is solid, it will be stated as fact. Where it is a working hypothesis, it will be identified as such. Where it is speculation grounded in plausible interpretation, it will be flagged as such. The science of ancient human genetics is a young field, still generating surprises. What is presented here reflects the current state of that science, with the understanding that new discoveries continue to refine and sometimes revise the picture. The story of this mutation is not a story with a single moment of dramatic change. It is a story about accumulation, small shifts in anatomy, in neural connectivity, in social behavior, in the complexity of what one human being could communicate to another. And it is a story that is still unfolding, because the mutation in question is present in every living human being on the planet today. You carry it. Every person you have ever met carries it. It was there when the first cities were built in Mesopotamia. It was there when the first boats crossed from Southeast Asia to Australia. It was there when someone first pressed a reed into wet clay and made a mark that stood for a word. The mutation preceded all of that. It made all of that possible, though it did not make any of it inevitable. Stay with this for a while. The standard account of human evolution tends to organize itself around anatomy. We track the gradual enlargement of the brain case. We note when stone tools appear in the archaeological record, and when they become more sophisticated. We mark the spread of our species out of Africa. We point to the cave paintings of Lascaux and Altamira as evidence that fully modern human cognition was in place by at least 35,000 years ago, probably earlier. This account is accurate as far as it goes, but it tends to treat language as something that appeared relatively late and relatively suddenly, as if one generation of early humans could communicate roughly the way chimpanzees do, and a few thousand years later their descendants were composing oral epics around fires. The reality was not like that. The capacity for complex human language is the product of multiple genetic changes, anatomical adaptations, and neurological reorganizations that accumulated over hundreds of thousands of years. FOXP2 is the most extensively studied of the genes involved, and it is the one that gives this story its sharpest focus, because the human version of FOXP2 differs from the chimpanzee version at exactly 2 amino acid positions, and those 2 differences appear to have been under strong positive selection at some point in human prehistory, meaning that individuals who carried the newer versions of the gene had significantly more children than those who did not. That kind of signal in the genome is the fingerprint of a change that mattered enormously to survival and reproduction. What did FOXP2 actually do? P2. The gene codes for a transcription factor, which is a protein that regulates the activity of other genes. It is active during fetal brain development and continues to influence the function of certain brain regions throughout life. The regions most affected include the basal ganglia, the cerebellum, and portions of the cortex involved in sequencing and learning complex motor patterns. In practical terms, a fully functional human version of FOXP2 supports the development of the fine motor control needed for speech, the ability to coordinate at very high speed the movements of the tongue, the lips, the jaw, the soft palate, and the muscles of the larynx. It also supports the neural circuits that allow the brain to learn and reproduce complex sequences of sound, and to associate those sound sequences with meanings. Mice with a disrupted version of FOXP2 produce abnormal vocalizations and have difficulty learning the motor sequences required for complex movements. Humans born with only one functional copy of FOXP2 have a characteristic pattern of difficulties. They struggle with articulating speech sounds, with understanding and producing grammatically complex sentences, and with certain fine motor tasks unrelated to speech. This condition is rare, and it was the families affected by it, first studied by the geneticist Simon Fisher and his colleagues in the early 2000s, that led researchers to identify FOXP2 as a gene with a central role in the human capacity for language. The discovery did not mean that FOXP2 is the language gene, a phrase that was used carelessly for a few years and then abandoned. Language is too complex a capacity to be reducible to a single gene, but FOXP2 is one of the most significant known genetic contributors to the biological infrastructure that makes language possible, and the human-specific changes to it appear to be genuinely ancient, having occurred somewhere between 500,000 and 2 million years ago, based on various calculations, with the most widely cited estimate placing the sweep of the human variants at somewhere around 500,000 years ago, perhaps during the period when Homo heidelbergensis was the dominant human form in Africa and Europe. She bent forward over the fire in the early morning and blew. The coals brightened. A thin column of smoke rose and flattened in the cool air. Around her, the other members of her group were beginning to stir. A child untangling itself from a sleeping adult, a young man sitting up and scanning the surrounding savannah with the automatic, unhurried attention of someone who has spent every night of his life outdoors in a landscape that contains predators. They communicated, all of them, in ways that were sophisticated by the standards of any other primate. They had calls for different categories of threat. They had signals for food type and direction. They had sounds that functioned as names, or something like names. But the architecture of what they could say was narrow. They could not describe what had happened yesterday to someone who had not been there. They could not explain the concept of a plan to someone who had not participated in forming it. They could not, in the sense that we would recognize, tell a story. Somewhere far to the northeast, the man with the changed gene was sleeping beside his own fire. He had no idea that his children, if he had them, and his children's children, and the long line descending from him across thousands of years, would carry something inside them that would eventually, in combination with other changes still to come, make possible the sentence you're listening to right now. Somewhere far to the northeast, the man with the changed gene was sleeping beside his own fire. He had no idea that his children, if he had them, and his children's children, and the long line descending from him across thousands of years, would carry something inside them that would eventually, in combination with other changes still to come, make possible the sentence you're listening to right now. The mutation did not announce itself. There was no visible change in the man who carried it. He looked like the other men in his group. His skull had the same prominent brow ridges, the same receding forehead, the same large teeth suited to a diet that required significant chewing. He was strong in the way that all the people of his time were strong, with a bone density and musculature that would strike a modern observer as extreme. He ran well and threw accurately and could read the behavior of animals with a precision that took years to develop. Nothing about him, from the outside, suggested that anything was different. The difference was internal, and it was subtle, and it expressed itself gradually across the years of his childhood and adolescence, in ways that were probably not obvious even to himself. When he was very young, learning to produce the calls and signals of his group, he may have learned them slightly faster than other children. The motor patterns required to shape sounds with the lips and tongue may have felt slightly more natural to him, slightly more available, the way a person with a good ear for music finds it marginally easier to reproduce a melody than someone without that particular advantage. The neural pathways supporting the fine motor control of his vocal tract were, because of the protein his FOXP2 variant produced during fetal development, wired with a slightly greater degree of precision and flexibility. Over the course of his life, this may have translated into a marginally larger repertoire of distinct calls, a slightly greater ability to modulate the acoustic properties of the sounds he produced, a slightly better capacity to combine signals in sequence in ways that carried more specific meaning. The difference between him and his companions would not have been the difference between language and non-language. It would have been the difference between a system with 40 distinct signals and a system with 55, between calls that could be combined in 3 ways and calls that could be combined in 8 or 9. That is not a dramatic difference. But in a world where communication determined how well a group could coordinate during a hunt, how effectively warnings could be transmitted, how precisely knowledge could be shared between an older person and a younger one, even a marginal advantage in communicative capacity translated into real differences in survival and reproduction. The children of people who communicated more effectively were more likely to live. The children of those children were more likely to live. Over many generations, the variant spread. This is the basic logic of positive selection, and it is the logic that the genome of every living human being records in the FOXP2 region. When geneticists compare the human version of this gene to the versions found in our closest living relatives, what they see is the signature of a selective sweep, a pattern in the surrounding DNA that indicates that a new variant rose from a very low frequency to near universal frequency in the population very rapidly, in evolutionary terms, because individuals who carried it left significantly more descendants than those who did not. The mathematics of such a sweep, given the estimated size of human populations in the relevant period, suggest the process took somewhere between 10,000 and 100,000 years. In geological time, that is almost instantaneous. In human terms, it is 3,000 to 5,000 generations of people living, reproducing, and dying without any awareness that anything was changing. It is worth understanding what the world looked like during the period when this selective sweep was most likely occurring. The best current estimates place the critical divergence of the human FOXP2 variants somewhere in the range of 500,000 to 700,000 years ago, though some analyses push components of the story earlier or later. This falls within the period that paleontologists associate with Homo heidelbergensis, a broad and somewhat contentious category that encompasses the archaic humans living in Africa, Europe, and possibly Western Asia during the Middle Pleistocene. These were large-brained people by any reasonable standard, cranial capacities ranging from about 1100 to 1300 cubic centimeters, overlapping substantially with the lower end of the modern human range. They made symmetrical stone tools, the Acheulean hand axes that required planning, and a degree of aesthetic judgment in their manufacture. There is evidence, though not conclusive evidence, that they used fire deliberately. There is evidence that they sometimes cared for injured or incapacitated group members over extended periods, which implies a level of social cohesion and reciprocal obligation that extended beyond immediate kin relationships. These were not simple animals. They were cognitively sophisticated creatures navigating complex social landscapes, tracking game through changing seasonal environments, and managing the practical challenges of life in Africa and Europe during a period of significant climatic instability. The ice ages were tightening their grip on the northern latitudes, and the oscillation between cold and warm periods was reshaping habitats and animal distributions on timescales of thousands of years. Groups that could plan, coordinate, and communicate more effectively had real advantages in this environment, and among those groups, the individuals who could communicate with slightly more precision and flexibility had real advantages within their groups. The spread of the FOXP2 variant was not a revolution. It was a ratchet. Each click of the ratchet was imperceptible in a single lifetime. Across 10,000 years, across 50,000 years, the accumulated effect was real. The average communicative capacity of the population was shifting, incrementally, towards something more powerful. A woman sits at the edge of a shallow lake in what will one day be the southern part of the East African Rift Valley. She is perhaps 400,000 years after the man with the changed gene, which means the variant he carried has long since spread through the population. Nearly everyone in her group carries it now. She's doing something that none of the people alive at the time of our earlier scene could have done. She's making a sound, a sustained vocalization, that has no immediate practical reference. She's not signaling food or danger or direction. She's producing a sequence of sounds that the other people in her group recognize as a reference to something absent, something that is not here right now, something that happened in the past or might happen in the future. The sounds are not yet language in the full modern sense. The grammar is primitive, the vocabulary is tiny, but the reference is displaced. She is talking about something that is not present. This is one of the most significant thresholds in the history of communication. Most animal signaling systems are tied to the immediate present. The alarm call of a vervet monkey refers to the predator that is here right now. The honeybee dance refers to the food source discovered within the last few hours. Displaced reference, the ability to communicate about things that are not present in time or space, is one of the foundational features of human language, and it requires a cognitive architecture that most animals simply do not have. It requires the ability to form a mental representation of an absent entity, hold that representation in working memory, and translate it into a communicative signal that another individual can decode. It requires, in short, a brain that FOXP2 and its downstream genetic effects help to build. She finishes her vocalization. The other people around the lake respond. Some of them seem to understand, or partially understand. One older woman makes a sound in response that appears to disagree or modify or add something. The exchange lasts perhaps 30 seconds. Then they go back to what they were doing, preparing animal hides, tending the fire, watching the water for any sign of the large crocodilians that also use this lake. What was communicated in those 30 seconds? We cannot know. But the fact that something was communicated about an absent referent, across a group of individuals who shared a conventionalized system of sounds with assigned meanings, is itself the thing that matters. The infrastructure was being built, not consciously, not deliberately, just by people doing what people do, talking or proto-talking about the things that occupied their attention, and selecting, generation by generation, for the variants that made doing so slightly easier and slightly more effective. The anatomy was changing to match the neural changes. This is one of the underappreciated aspects of the FOXP2 story. The gene does not only affect the brain, it also influences the development of the orofacial structures, the muscles and bones of the face, mouth and throat that are involved in producing speech sounds. The human vocal tract is anatomically unusual among primates. The larynx sits lower in the throat than in any other ape, which creates a larger resonating chamber above it and makes possible a much wider range of vowel sounds. The tongue is more muscular and more flexibly mobile. The lips have greater independent motor control than in any other primate. The fine motor cortex, the strip of neural tissue running across the top of the brain that maps to the muscles of the body, devotes a disproportionately large area to the face, lips, tongue, and throat in modern humans compared to other primates. These are not features that appeared fully formed at a single moment. They accumulated. They were shaped partly by genetic changes, among them the changes driven by the human FOXP2 variant and the genes regulated by the FOXP2 protein, and partly by the feedback loop between anatomy and use. A throat that could produce more sounds was used to produce more sounds, and the populations and lineages in which that happened left more descendants. There is also the question of the brain regions downstream of FOXP2. The gene is expressed most heavily in the striatum, the part of the basal ganglia involved in learning and executing sequences of movement, and in the cerebellum, which coordinates movement timing and precision. Both of these regions have expanded and reorganized significantly in the human lineage compared to other apes. The striatum in particular is now understood to be deeply involved not just in motor learning but in language processing. It helps the brain sequence the sounds and grammatical elements of speech in the correct order, at the very high speeds that normal conversation requires. When the striatum is damaged, as it is in certain movement disorders, speech often becomes slow, laboured, and dysrhythmic. The human striatum has been shaped partly by the same genetic changes that shape the human vocal tract, and FOXP2 is a significant node in that network of influence. The man who had been following the herd for 6 days finally stopped on the morning of the 7th day, at the crest of a low ridge that overlooked a wide, shallow valley. The herd was below him, moving in loose formation across the pale grass toward a line of acacia trees that marked the presence of water. He watched them for a long time. Then he produced 3 sounds in rapid sequence. The 2 men with him understood immediately. They separated, moving quietly down either side of the ridge. The coordination required no further communication. The sounds he had made were not elaborate, but they were specific, and they were understood, and 3 people moved in 3 different directions toward a common goal because of them. That is language at its most compressed and functional. 3 sounds, 3 movements, 1 coordinated outcome. It does not look impressive from the outside. From the inside of evolutionary history, it is one of the most important things that has ever happened on this planet. To understand what the mutation built, it helps to understand what existed before it. The communication systems of the great apes are real, and they are not trivial. Chimpanzees in the wild use a repertoire of calls, gestures and facial expressions that carry genuine information. They have distinct alarm calls for aerial predators and terrestrial predators. They use gestures to direct attention and to request actions from other individuals. They engage in deliberate deception, which requires a model of what another individual knows and believes. They learn tool use by observation and transmit certain tool use behaviors across generations within specific communities, which is a rudimentary form of culture. Bonobos, their closest relatives and ours, show similar capacities and in some respects appear even more flexible in their use of gestural communication. Gorillas and orangutans have their own repertoires, their own social communication systems, their own learned behaviors. What none of them can do is produce the continuous, rapid, voluntarily modulated stream of sounds that constitutes human speech. This is not primarily a cognitive limitation. It is partly anatomical and partly neural. The larynx of a chimpanzee sits high in the throat, close to the base of the skull, in a position that allows the animal to breathe and swallow simultaneously, which is a useful safety feature, but which severely limits the range of sounds the vocal tract can produce. The tongue of a chimpanzee is long and flat, well suited to food manipulation, but not to the rapid, precise shaping of airflow that produces the consonants and vowels of speech. The motor cortex of a chimpanzee does not devote a disproportionate area to the face and throat the way the human motor cortex does, and the neural circuits linking the motor cortex to the muscles of the vocal tract. The circuits that allow a human speaker to produce 15 to 20 distinct sounds per second with sub-millisecond precision are organised differently in chimpanzees than in humans. Some of these differences are downstream of FOXP2. The chimpanzee version of the gene differs from the human version at 2 amino acid positions that appear to matter for how the protein interacts with the DNA of the genes it regulates. When researchers engineered mice to carry the human version of FOXP2 in place of the mouse version, the mice showed measurable changes in the structure and function of their corticostriatal circuits, the connections between the cortex and the striatum, and they learned certain motor sequences faster than control mice. They also showed changes in their vocalizations, the ultrasonic calls that mouse pups make when separated from their mothers. The changes were subtle and the mice did not begin producing human speech, but the experiment confirmed that the 2 amino acid differences between human and chimpanzee FOXP2 have real, measurable effects on neural development and function. This matters because it anchors the story in something concrete. The FOXP2 story is not purely theoretical. It is not a narrative constructed from indirect inference alone. It is supported by experimental evidence from multiple directions, genetic comparisons across species, clinical observations of humans with FOXP2 mutations, neuroimaging of healthy humans performing language tasks, developmental studies of how FOXP2 expression shapes the growing brain, and the engineered mouse experiments just described. The picture that emerges from all of these converging lines of evidence is consistent and coherent, even if many of the details remain under active investigation. One of those details concerns timing. When exactly did the human-specific FOXP2 variants arise and spread? The original estimate, based on the pattern of genetic variation surrounding the FOXP2 gene in modern human populations, suggested the selective sweep occurred roughly 200,000 years ago, which would place it close to or slightly before the appearance of anatomically modern humans in the fossil record. Later analyses have pushed this estimate back considerably, some to 500,000 years or more. The debate is not fully resolved. What is clear is that the human FOXP2 variants were present in Neanderthals and Denisovans, the archaic human lineages that diverged from our direct ancestors perhaps 500,000 to 800,000 years ago. This was confirmed when researchers sequenced the genomes of Neanderthal individuals from several European cave sites and found that their FOXP2 gene carried the same 2 human-specific amino acid substitutions found in all living people today. This finding was significant in ways that extended beyond the genetics. If Neanderthals carried the same FOXP2 variants as modern humans, then those variants predated the divergence between the Neanderthal and modern human lineages, and it suggested, though it did not prove, that Neanderthals may have had at least some of the biological infrastructure for language. This possibility is still debated, and it connects to a broader set of questions about Neanderthal cognition and communication that the physical evidence has not fully resolved. A Neanderthal woman in a cave in what will one day be called the Iberian Peninsula, roughly 50,000 years ago, is working on a piece of red ochre with a small stone tool, grinding it to powder on a flat limestone surface. She's not the only one doing this. 2 others are nearby, a young man and an older woman, both occupied with their own tasks. The red powder accumulates on the stone. She's been doing this for a long time and she will do it again tomorrow and the day after. The ochre is used for something, though exactly what is debated. Body decoration, hide preservation, symbolic marking of objects or surfaces. The point is that its use is deliberate, its preparation is effortful, and it is being done by 3 individuals in proximity without apparent conflict or confusion about what each is doing. This is a scene of social coordination at a level that goes beyond what most animals achieve. The individuals in that cave are not just tolerating each other's presence. They are engaged in related activities in a shared space, with some common understanding of purpose. Whether they communicated about that purpose in anything resembling language is unknown, but they carried the FOXP2 variants. Their vocal tracts, as far as we can tell from the anatomy of the hyoid bones and the geometry of the base of the skull, were not identical to modern human vocal tracts, but were closer to them than to chimpanzee vocal tracts. The hyoid bone, the small horseshoe-shaped bone at the base of the tongue that anchors many of the muscles used in speech, was recovered from a Neanderthal site at Kibara Cave in Israel, and its morphology is remarkably similar to that of modern humans. The geometry of the base of the skull in Neanderthal fossils has been analysed using 3-dimensional modelling, and the results suggest a vocal tract capable of producing a substantial range of vowel sounds, though perhaps not the full modern human range. The evidence does not allow us to say that Neanderthals spoke language as we understand it, but the evidence does not allow us to say they did not either. What it suggests is that they had a significant portion of the biological machinery that makes language possible, and that this machinery was present in them because they shared the genetic changes that built it, including the changes in FOXP2, with the common ancestor of Neanderthals and modern humans. The Denisovans present an even more interesting case, and a more frustrating one, because what we know about them comes almost entirely from ancient DNA, rather than from a substantial fossil record. The Denisovans were identified as a distinct human lineage in 2010, when researchers at the Max Planck Institute for Evolutionary Anthropology sequenced DNA extracted from a finger bone found in Denisova Cave in the Altai Mountains of Siberia. The genome that emerged from that analysis was clearly human, but clearly distinct from both modern humans and Neanderthals. The Denisovans had diverged from the Neanderthal lineage perhaps 300,000 to 400,000 years ago, which places their divergence well after the origin of the human FOXP2 variants. Their FOXP2 sequence, as far as it can be reconstructed, also appears to carry the human-specific variants. They too, then, were organisms whose vocal development was shaped by the same genetic changes that shaped ours. What we do not know about the Denisovans is profound. We have a few teeth, a finger bone, a toe bone, and a jaw fragment found in Tibet. We have their genome, which is extraordinarily informative about their relationships to other human lineages, but tells us relatively little about what they looked like, how they lived, or what they were capable of communicating. The Tibetan jaw fragment, analysed in 2019, suggests they were physically robust, with large teeth suited to a demanding diet. The altitude of the site suggests they were adapted to high-altitude environments, and their genome shows variants in genes related to hemoglobin oxygen binding efficiency that are found in modern Tibetans, who appear to have inherited them through ancient interbreeding with Denisovans. She sets down the grinding stone and looks at the powder on the flat surface. It is the right colour now, a deep reddish brown. She picks up a small piece of folded hide and presses it carefully against the powder, lifting some of it onto the surface of the hide. The older woman nearby makes a sound. The younger woman looks up and responds with a sound of her own. The exchange lasts perhaps 4 seconds. Then both of them look back at what they are doing. Something was communicated. We will never know what, but both individuals adjusted their behaviour in the seconds following the exchange, which means information moved from one nervous system to another through voluntary sound production. Whatever that was, it was part of the same long continuum of capability that FOXP2 helped to build in Neanderthals and in us, and in the shared ancestor before either lineage existed. The question of what distinguishes modern human language from whatever Neanderthals and Denisovans may have had is real, and it connects to questions that go beyond FOXP2. FOXP2 is one piece of the genetic story, but the genetic architecture of language is vastly more complex than a single gene, and the parts of that architecture that appear to be unique to anatomically modern humans, those that may have given our lineage a communicative edge over our archaic relatives, involve many other genes and many other changes. Among those changes, some of the most interesting involve not the coding sequences of genes, those portions of the DNA that specify the structure of proteins, but the regulatory sequences that control when, where, and how strongly a gene is expressed. Modern humans differ from Neanderthals and Denisovans in hundreds of positions in the regulatory regions of genes that are active in the developing brain and vocal tract. Some of those differences may have fine-tuned the language-related circuitry in ways that gave modern humans capabilities their cousins lacked. The young man in the cave had been quiet for a long time. He was watching the entrance, where the late afternoon light was beginning to angle in at a low orange slant. He made a sound, low and brief. The woman with the ochre paused. The older woman set down what she was holding. All three of them listened. From outside came the distant sound of movement in dry brush, something large moving without particular care for silence. They held still for perhaps 10 seconds. Then the sound moved away, tracking northward along the base of the cliff. The young man made a different sound, one that clearly indicated the threat had passed, and the two women went back to work. 3 sounds, 3 responses, a coordinated adjustment of behaviour in response to shared information. In that cave in Iberia 50,000 years ago, something very much like language was probably at work, built on the same biological foundation that FOXP2 helped to lay, expressed in bodies that were not quite like ours, but were far closer to ours than to any other animals. And the story of FOXP2 is inseparable from the story of the throat. Most people, if asked to identify the most important organ for language, would say the brain. They would not be wrong, but the brain's contribution to language is invisible from the outside. What is visible, what is audible, is the sound that comes out of the body, and the production of that sound depends on a set of anatomical structures that are, in the human species, configured in a way that is genuinely unique among all living primates. The larynx, sometimes called the voice box, sits in the throat and houses the vocal cords, two folds of mucous membrane stretched across a cartilaginous frame. When air passes through the larynx during exhalation, the vocal cords can be brought together so that they vibrate, producing a buzzing tone that the rest of the vocal tract then shapes into recognizable sounds. In chimpanzees and other great apes, the larynx sits high in the throat close to the base of the skull. This high position allows the soft palate at the back of the mouth to contact the top of the larynx during swallowing, creating a seal that keeps food and liquid out of the airway. It is a good design for an animal that climbs trees and needs to breathe easily while chewing and swallowing. But it severely limits the resonating space above the vocal cords, and that space, the pharynx, is what determines the range of vowel sounds an animal can produce. In adult modern humans, the larynx is descended to a position roughly in the middle of the neck. This lowered position creates a long, flexible pharyngeal cavity above the vocal cords, and that cavity can be reshaped by the movements of the tongue and the walls of the throat to produce the wide range of vowel sounds that are the acoustic backbone of human speech. It also creates a vulnerability. Because the airway and the food passage cross at the level of the pharynx in adult humans, there is a genuine risk of choking, a risk that does not exist in the same way for other apes. Evolution accepted this vulnerability as the price of a more powerful vocal tract. The descended larynx does not appear fully in human infants at birth. Newborn humans have a larynx positioned relatively high in the throat, similar to the ape configuration, which allows them to breathe and nurse simultaneously. Over the first year and a half of life, the larynx gradually descends to its adult position, a process that coincides with the period of rapid speech development. By the time a child is 18 months old and beginning to produce two-word combinations, the larynx is already most of the way to its adult position. The correlation is not coincidental. FOXP2 does not directly specify the position of the larynx. The anatomy of the vocal tract is the product of many genes and many developmental processes, but FOXP2 regulates genes that are active in the tissues of the head, neck, and throat during fetal development, and disruptions to FOXP2 function produce anomalies not just in neural circuitry, but in the development of orofacial structures. The gene sits at an intersection between brain development and body development, influencing both the neural machinery that controls speech and the physical machinery that produces it. A child is sitting near a fire somewhere in the southern African interior, perhaps 90,000 years ago. He's approximately 2 years old. He has recently begun producing strings of sounds that the adults around him recognize as meaningful. Not just single calls, but sequences, 2 or 3 distinct sound units combined in ways that refer to specific things and situations. He's doing what human children everywhere and in every era have done. He's learning language by immersion, by listening, by attempting to reproduce what he hears, by receiving feedback from the people around him when his attempts succeed or fail. The process looks unremarkable because it is unremarkable in the sense that it has been happening continuously in human populations for hundreds of thousands of years. But it is also one of the most astonishing things that happens in the animal world. A 2-year-old organism acquiring rapidly and apparently effortlessly a complex symbolic communication system without formal instruction, guided only by exposure and the powerful drive to communicate that seems to be built into the human nervous system from birth. The ease with which human children acquire language is itself one of the most powerful arguments for the deep genetic basis of the capacity. Children do not learn to walk by being taught in the way that one teaches a skill. They develop the ability to walk as the relevant neural circuits mature and the relevant muscles strengthen, and they practice obsessively because something in their nervous system drives them toward upright locomotion. Language acquisition is similar. Children do not need to be formally taught grammar. They extract the grammatical rules of whatever language they are exposed to from the raw input of the speech they hear, inferring complex regularities from a data stream that would defeat any computer algorithm working without a prior model of how human grammars are structured. This has led linguists, most famously Noam Chomsky, to propose that the human brain comes equipped with a language acquisition device, a set of innate constraints and biases that make certain kinds of grammatical structures easy to learn and certain others essentially impossible. Whether that proposal is correct in its strong form is still debated, but the extraordinary speed and uniformity of language acquisition across cultures and across children with very different levels of language exposure argues powerfully for a substantial innate component. That innate component has a genetic basis, and among the genes that contribute to it, FOXP2 is the best documented and the most ancient. Its contribution is not to grammar in the abstract sense. It does not encode the rules of syntax or the principles of semantic composition. Its contribution is to the neural and physical infrastructure that makes the production and perception of complex speech sounds possible, and to the circuits that allow the brain to learn and reproduce long, precise sequences of motor acts at high speed. Without that infrastructure, the abstract machinery of grammar has nothing to run on. FOXP2 is not the architect of language. It is one of the engineers who built the building that the architect later filled. The child by the fire produces a string of 3 sounds and looks at the woman beside him with an expression of unmistakable expectation. She responds. He produces the string again, slightly differently. She responds again with a slight modification of her own sounds. This exchange, which lasts less than a minute, is a language lesson of a kind that has been taking place for tens of thousands of years. An older speaker and a younger one calibrating their shared system, adjusting toward convergence, building the common code that will allow them to communicate with precision across their lifetimes. Outside the range of the firelight, the savannah is dark and full of sound. Insects in the grass, the distant call of a night bird, the occasional low sound of large animals moving somewhere to the west. The people around the fire pay attention to all of it, without appearing to pay attention. This is the other side of the communication equation: reception. The neural circuits that process incoming sound in modern humans are also shaped by FOXP2 and the downstream genes it regulates. The auditory cortex of modern humans is specialized not just for detecting and discriminating sounds, but for processing speech-specific features, the rapid formant transitions that distinguish one vowel from another, the voice-onset timing differences that separate voiced consonants from voiceless ones, the prosodic patterns of stress and intonation that carry information about sentence structure and speaker intent. This auditory specialization is part of the same system that FOXP2 helped to build. It develops in parallel with the motor system for speech production, and the two systems are linked by the neural circuits of the arcuate fasciculus, a large fiber bundle that connects the posterior temporal lobe, where speech is primarily processed, to the frontal lobe, where it is primarily planned and produced. The arcuate fasciculus is one of the neural features that distinguishes humans most sharply from other primates. In chimpanzees, the connections between the temporal and frontal lobes are much sparser than in humans, which may be part of why chimpanzees, despite having substantial cognitive abilities, cannot learn to produce speech even when raised from infancy in human households and exposed to spoken language continuously. The bottleneck is not comprehension, which chimpanzees can achieve to a surprising degree in various experimental formats. The bottleneck is production, the ability to convert a desired communicative intention into a precisely timed sequence of muscle movements in the vocal tract. That conversion requires the dense corticobulbar pathways, the direct connections from the motor cortex to the brainstem motor neurons that control the muscles of the face and throat that are present in humans and largely absent in chimpanzees. FOXP2 influences the development of these pathways. Along the shore of a lake that no longer exists, in a landscape that is now dry scrubland in northern Tanzania, a group of perhaps 15 people is doing something that would have been impossible without the genetic changes that preceded them by hundreds of thousands of years. They are engaged in what can reasonably be called a discussion. Several individuals are producing extended vocalizations in sequence, waiting for others to finish before producing their own, occasionally producing overlapping sounds that appear to function as acknowledgements or affirmations. The topic of the discussion, if it can be called that, appears to concern a decision about where to move next, a set of signals referring to directions, distances, the presence or absence of water, the likely behavior of the animals they depend on. This is deliberative communication, Not just the transmission of a signal and the reception of a response, but the joint construction of a shared understanding about a complex situation through multiple rounds of exchange among multiple participants. This is something that no other animal does in quite this way. Wolves coordinate hunts through a combination of body language and vocalization, but there is no evidence that wolves deliberate about strategies before a hunt begins. Elephants have rich communication systems and strong social bonds, and their vocalizations carry substantial information about individual identity, emotional state, and social relationships. But elephant communication, as far as we understand it, does not support the kind of multi-party, multi-round deliberative exchange that those 15 people by the lake were engaged in. The gap is not absolute. It is a matter of degree, of complexity, of the density of information that can be transmitted and processed. But the gap is real, and FOXP2 is part of what produced it. One of the people in that group by the lake is an older man who has not been part of the main exchange. He has been listening, occasionally making small sounds that function like acknowledgements, mostly watching the faces of the others. He has survived to an age that, in his world, represents a significant accumulation of experience. He knows things that the younger people in the group do not know, including things about this specific landscape that they have not yet encountered and could not derive from first principles. And when the main exchange pauses, he produces a sustained vocalization that lasts perhaps 20 seconds. The others listen. Some of them respond briefly. Then there is a period of quiet, after which the group begins to move, organizing itself with the ease of a coordinated decision in a direction that the old man had indicated. Knowledge transferred, decision made, group moved, all because of sounds shaped by a throat whose architecture had been centuries in the building, processed by brains whose circuits had been laid down by genes whose history stretched back further than any of them could imagine. Language did not arrive alone. It came with a set of cognitive changes that are difficult to separate cleanly from the communicative changes because the two are so deeply intertwined. The ability to produce complex sequences of sounds with precise timing is related, at the neural level, to the ability to plan and execute complex sequences of actions more generally. The same corticostriatal circuits that FOXP2 helped to build, the circuits connecting the frontal cortex to the basal ganglia, are involved not just in speech production, but in all forms of sequential motor learning, in planning, in the flexible switching between behavioral strategies, and in the ability to suppress an automatic response in favor of a deliberate one. When those circuits changed, language changed, but behavior changed too, in ways that were not strictly linguistic. This is one of the reasons that researchers studying the archaeological record of behavioral modernity, the cluster of traits that appear to distinguish fully modern human behavior from the behavior of earlier hominins, have found it so difficult to draw clean boundaries. The traits cluster together. Symbolic behavior, the use of ochre and shell beads as personal ornament, tends to appear in the same assemblages as evidence of long-distance exchange networks, which require the ability to maintain social relationships across distances and to communicate about obligations and reciprocity. Complex composite tool manufacture, the gluing of stone points onto wooden handles using compound adhesives, requires a degree of planning and sequential precision that overlaps with the planning demands of complex speech. The production and maintenance of symbolic objects implies a shared representational system, a set of meanings that exist by convention and that must be communicated and transmitted across generations. None of these behaviors require language in the way that, say, a specific grammatical construction requires a specific neural circuit, but all of them are facilitated by language, and all of them tend to appear in the archaeological record in contexts where language, based on the available anatomical and genetic evidence, was likely present. The period between roughly 100,000 and 40,000 years ago is the one that paleoanthropologists and archaeologists find most interesting in this regard, because it is the period during which the archaeological signature of modern human behavior becomes most clearly visible and most clearly distinct from what came before. This period is associated with the Middle Stone Age in Africa and, from around 50,000 years ago, with the Upper Paleolithic in Europe and Western Asia. It is the period when personal ornaments first appear systematically, when evidence of deliberate pigment use becomes widespread and consistent, when the first unambiguous examples of representational art appear, and when the pace of technological innovation begins to accelerate in a way that has no precedent in the prior 2 million years of hominin history. A man is sitting at the back of a shallow rock shelter in the Cape region of southern Africa, approximately 75,000 years ago. The shelter faces east, catching the morning light. He's working on a piece of ochre about the size of his fist, abrading its flat surface against a coarser stone to produce a thin film of red powder. He collects the powder carefully, using a small folded piece of hide as a scraper. He is not alone. There are 7 other people in the shelter, in various states of activity, sleeping, preparing food, tending a small fire near the entrance. He works steadily and without apparent urgency, and when he has collected enough powder, he sets the ochre block aside and picks up a small perforated shell from a collection of perhaps 20 such shells near his right knee. He threads a thin strip of hide through the perforation and ties a careful knot. Then another shell, then another. He is making something that will be worn, something that will be visible on a body, something that will communicate information about the person wearing it to anyone who sees it. This scene, or something very much like it, is recorded in the physical evidence from sites along the southern coast of Africa, most extensively from the site known as Blombos Cave on the southern Cape coast of South Africa. Blombos has produced perforated shells that functioned as beads, pieces of ochre with engraved geometric patterns, and a collection of small stone tools of a sophistication that places them at the forefront of Middle Stone Age technology. The engraved ochre pieces are particularly significant. They are flat slabs of red ochre whose surfaces bear carefully incised crosshatch and geometric patterns. These patterns are not accidental. They were made deliberately with precision by someone who had a clear idea of what they wanted to produce before they began producing it. They communicate nothing that we can decode, but the fact that they were made at all, that someone in southern Africa 75,000 years ago put deliberate symbolic marks on a piece of red stone, is a piece of behavioral evidence that speaks to the cognitive world of its maker. That cognitive world was one in which symbols existed, in which a physical object could carry a meaning beyond its material properties, in which the meaning of a mark or an ornament was shared between maker and viewer through convention, through a code that had to be learned and transmitted. That is the same world that language lives in. Every word is a conventional symbol. The sequence of sounds that constitutes the word water has no inherent relationship to the liquid it refers to. The relationship is arbitrary and conventional, established by use and maintained by transmission. The shells and the engraved ochre and the geometric marks belong to the same cognitive universe as the arbitrary sound-meaning relationships of language. A universe in which the mind can create symbols and share them with other minds through learned convention. FOXP2 did not create the capacity for symbolic thought. Symbolic thought has a neural basis that extends well beyond the circuits that FOXP2 influences. But the same selective pressures that favoured individuals with better vocal motor control also favoured individuals with better symbolic cognitive abilities, because language and symbolic cognition are part of the same adaptive complex. They emerged together, reinforced each other, and spread together through the populations that carried the genetic changes enabling them. The man making shell beads in the rock shelter was living in a cognitive world that his ancestors 500,000 years earlier could not have inhabited, and the genetic changes in FOXP2 and in many other genes were part of what made that world accessible to him. The rock shelter by the sea holds another layer of significance that is worth dwelling on. The southern coast of Africa during periods of glacial maximum, when ice sheets locked up enormous quantities of water and global sea levels dropped by as much as 120 meters below present levels, was a dramatically different landscape from what it is today. The continental shelf extended much further south, creating a broad coastal plain with sheltered bays, wetlands, and rich intertidal zones. The climate along this coast remained relatively mild even during glacial maxima, buffered by the ocean. This may have made the southern African coast one of the refugia, where anatomically modern human populations survived and maintained their numbers during periods when much of the continent was becoming increasingly arid and inhospitable. If that is correct, then some of the people living in caves like Blombos 75,000 years ago were not just behaviourally modern in the sense of making beads and engraving ochre. They were the descendants of a population that had been relatively isolated and relatively stable for many thousands of years, maintaining a lineage in which the genetic changes contributing to modern cognition and language had been accumulating and being refined. When the climate improved and populations expanded again, the descendants of those coastal people would carry with them not just genes but cultural knowledge, the shell bead tradition, the ochre working tradition, the tool-making traditions that had been preserved and developed through the long cold centuries. That cultural knowledge was transmissible only because language was there to transmit it. A girl of perhaps 12 years is watching the man with the shells. She has been watching him for a long time, quietly, from a distance of about 2 meters. She's not being formally taught. He's not demonstrating anything for her benefit. But she's learning, in the way that children of her time at all times learn by watching skilled adults, absorbing the sequence of actions, the selection of materials, the handling of the tools, the final judgment about when something is finished and right. She will be able to make shell beads herself within a year, not because anyone will sit down and instruct her step by step, but because she will have watched enough, and at some point she will pick up a shell and a piece of hide and try, and the trying will activate the motor learning circuits that her Fox P2 variant helped to build, and within 100 attempts she will have the skill. But something else is also happening. As she watches, the man occasionally produces sounds, brief quiet vocalizations that are not directed at her, but that accompany his work in the way that people sometimes talk to themselves, or narrate what they are doing in a low voice. She catches some of those sounds. They refer, she understands dimly, to the quality of the shell, to the placement of the perforation, to the tightness of the knot. The sounds carry information that the watching alone does not carry. The watching shows her what to do. The sounds, when she can pass them, begin to explain why. This is the cognitive advantage of language in cultural transmission. It does not replace learning by observation, but it adds a layer of explanation, of causation, of principle that makes the transmission faster and more robust and more accurate. A skill transmitted by observation alone tends to drift over generations, accumulating errors the way a photocopy of a photocopy accumulates degradation. A skill transmitted by observation plus language explanation can be corrected, clarified, and maintained with much higher fidelity. This is one of the reasons that modern human cultures evolved so much faster than any earlier hominin culture. The Acheulean tool tradition, the symmetrical hand axes made by Homo heidelbergensis and earlier hominins, persisted for over 1 million years with relatively little change. The same basic design, the same basic technique, generation after generation after generation. The pace of change was glacial in every sense. The Upper Paleolithic, by contrast, saw innovation at a pace that had no precedent. New tool forms appearing, spreading, and being replaced within thousands of years rather than hundreds of thousands. Representational art appearing and developing, bone and antler working, tailored clothing, long-distance trade in prestige materials. The difference is not primarily one of raw intelligence. It is primarily one of transmission fidelity and cumulative cultural learning, and both of those capacities depend on language. She picks up one of the finished shells from the collection near the man's knee. She holds it up to the morning light coming through the shelter entrance, turning it slowly. The perforation is clean and precise, made without breaking the shell. She sets it back down carefully and returns to watching. Somewhere in her developing brain, the circuits for sequential motor learning and for language comprehension are both active, both recording, both preparing her for the moment when she will pick up a shell of her own and begin. The genetic story of Fox P2 does not end with the mutation itself. It extends outward into a network of genes that Fox P2 regulates, genes that are themselves involved in brain development, neural connectivity, and the fine-tuning of the circuits that support language and complex cognition. When researchers mapped the full set of genes whose activity is influenced by the Fox P2 protein, they found a network of hundreds of targets, many of them involved in the development of the striatum, the cerebellum, and the cortex. Some of those targets are genes whose variants have independently been associated with language disorders, autism spectrum conditions, and other developmental conditions that affect communication. This network view of Fox P2 is more accurate than the single gene view, and it is more informative about how the mutation actually worked its effects on the brain and body of the people who carried it. Among the most interesting downstream targets of Fox P2 is a gene called CNTNAP2, which codes for a protein involved in the clustering of potassium channels in neurons, a process that affects the speed and reliability of neural signal transmission. CNTNAP2 is one of the largest genes in the human genome, and it contains regulatory regions where the FOXP2 protein binds and influences expression, Variants in CNTNAP2 have been associated with specific language impairment, with autism, and with epilepsy. The gene is expressed predominantly in the frontal lobe during fetal brain development, the region of the cortex most heavily involved in planning, sequencing, and the higher order processing of language. When FOXP2 changed in the human lineage, its effects cascaded through CNTNAP2 and through many other downstream targets, reshaping the developmental trajectory of the frontal lobe and the circuits connecting it to the rest of the brain. This cascading effect is part of what makes FOXP2 so significant as a node in the genetic network of language. A transcription factor that regulates hundreds of other genes is not just one piece of the puzzle, it is a master switch that, when it changes, alters the expression of a large portion of the developmental program of the brain. The 2 amino acid changes in the human FOXP2 protein affect how tightly and how specifically it binds to the regulatory regions of its target genes. Even small changes in binding affinity can translate, during the critical periods of fetal brain development, into measurable differences in the relative growth of different brain regions, the density of synaptic connections in particular circuits, and the timing of developmental events that determine the final architecture of the adult brain. In the highlands of what will one day be Ethiopia, roughly 60,000 years ago, a woman is giving birth. It is her second child. The first, a boy of 3 years, is sitting a short distance away in the care of an older woman who is not his mother. The birth is proceeding in the way that human births have always proceeded, with difficulty and with assistance. Unlike the births of other great apes, human birth requires help. The infant's head is large relative to the birth canal, a consequence of the rapid expansion of the brain case that occurred over the preceding 2 million years of human evolution. The attending woman knows what to do. She has assisted at births before. She positions herself in front of the labouring woman, ready to guide the infant as it emerges, turning it gently to navigate the curve of the birth canal. She does not know anatomy in any formal sense. She knows what works, transmitted through generations of women who attended births and talked about them afterward, passing on the knowledge of position and timing and when to apply gentle traction and when to wait. That last part matters. They talked about it. The knowledge of how to assist a human birth was preserved and transmitted, in this population and in populations like it across the continent, partly through language. The older woman attending this birth learned what she knows from an older woman who attended her own births, who learned from a woman before her. Each generation added what it observed and corrected what did not work, and the knowledge was more accurately preserved because it was encoded in words, in descriptions of what to do and what to watch for, and not only in the demonstration of hands that the next generation watched and imitated. Language as a vehicle for medical knowledge is not a modern invention. It is one of the oldest applications of the human communicative capacity. In a world without writing, without any external storage medium for information, the only way knowledge survived across generations was in the minds of living people, and the only way it was reliably transferred from one mind to another was through speech. The accumulated medical knowledge of a group, the understanding of which plants treated fever, which injuries could be splintered and which could not, how to manage the complications of childbirth, how to recognize the signs that an infection was becoming dangerous, all of that knowledge lived in language. It was generated by observation and experience, organized into communicable form by the minds that had acquired it, and transmitted to the next generation through the one technology that all human beings carry with them, the ability to speak. The infant is born. It is a girl and she is healthy, and she cries immediately, which is the sound that indicates the lungs are working. The attending woman cuts the umbilical cord with a sharp stone flake and wraps the infant in a folded hide that has been kept warm near the fire. The mother, exhausted, holds her daughter against her chest. The boy of 3 years approaches cautiously and is allowed to look. The older woman produces a sustained, low vocalization that everyone present understands as an expression of a particular kind of satisfaction. The specific relief of a dangerous thing successfully concluded. Everyone in the small group, the 4 adults and 2 older children who have been waiting nearby, understands immediately. They know without being told that the birth went well, and they adjust their own emotional states accordingly, relaxing in a way that is visible in their posture and their faces. That collective emotional adjustment mediated by a vocalization is one of the less obvious but deeply important functions of language in human social life. Language does not only transmit factual information, It transmits emotional and social information. It coordinates the emotional states of a group. It synchronizes the collective attention of individuals around shared concerns. Laughter is a form of this. Mourning is a form of this. The low communal murmur of a group settling down to sleep is a form of this. All of these vocalizations do work that goes beyond the propositional content of any individual utterance. They maintain the social fabric. They signal belonging and exclusion. They regulate the level of alert or relaxation in a group. And they are possible only because the neural architecture that FOXP2 and its network help to build is capable of producing not just precise, rapid articulations of referential content, but also the prosodic variations of pitch, timing, and intensity that carry emotional meaning. The prosody of speech, the music of it, the rise and fall of pitch that distinguishes a question from a statement, the slowing and softening that signals intimacy or grief, the quickening and sharpening that signals urgency or danger, is processed in different neural regions than the propositional content. The right hemisphere of the brain is more heavily involved in prosodic processing, while the left hemisphere dominates in the processing of syntactic and semantic content in most right-handed people. Both hemispheres are part of the language system. Both were shaped by the evolutionary changes that FOXP2 contributed to. The full communicative power of human speech depends on both channels working together, the words carrying the what and the prosody carrying the how and the why. The girl grows. She is, by the standards of her time and place, an ordinary child. She talks in the way that children of her era talked, early, eagerly, and with a comprehension that often outpaces her production. By the time she is 4 years old, she's constructing sentences of 5 and 6 words, and using them to ask questions about things that are not immediately present, questions about where things are, about what happened, about what will happen. She is doing something that no chimpanzee raised in a human household, exposed from birth to continuous spoken language, has ever done. She is spontaneously producing grammatically structured sentences in a human language. The capacity is in her. It was put there by the long sequence of genetic changes that preceded her, by the developmental program that those changes set in motion, by the neural circuits that grew during the months she spent in the womb, and the years she spent learning to walk and talk. The question of where language ultimately came from, of the first moment when the communicative system of a human or proto-human population crossed some threshold and became recognizable as language in the full modern sense, is one that science has not answered and may never fully answer. Language does not fossilize. The sounds spoken around fires 300,000 years ago are gone completely. What survives are the bones, the stones, the pigments, the shells. From those things we can reconstruct aspects of behavior, and from behavior we can infer aspects of cognition, and from cognition we can reason about communication. But the inference chain is long, and each link introduces uncertainty. What we can say with confidence is this: the genetic changes that supported the development of complex language were in place, in some form, well before the appearance of anatomically modern humans in the fossil record. The FOXP2 variants that all living humans carry were present in Neanderthals and Denisovans as well, implying that the common ancestor of all these lineages, living perhaps 600,000 to 800,000 years ago, already carried them. The anatomical changes to the vocal tract that support the production of complex speech sounds appear to have been accumulating over a similar timescale, judging from the morphology of fossil skulls and hyoid bones. And the behavioural evidence from sites like Blombos Cave suggests that by 75,000 years ago, at least some populations of modern humans were living in a fully symbolic cognitive world, making and wearing personal ornaments, engraving geometric patterns, maintaining long-distance exchange relationships, and very likely talking about all of it in ways that we would recognise as language. The network of genes downstream of FOXP2, including CNTNAP2 and the hundreds of other regulatory targets, was not static after the initial selective sweep. Evolution does not stop. The populations that carried the human FOXP2 variants continue to accumulate additional mutations, some of which further refined the language-related circuitry, some of which had no effect, and some of which occasionally were disadvantageous and were removed by selection. The genome is always in motion. The FOXP2 story is a chapter in a longer story of continuous genetic change, a story that began long before FOXP2 and has continued to the present day. The older woman who assisted at the birth has moved away from the fire and is sitting at the edge of the group looking out over the darkening highland plateau. The air is cooler here than on the coast, and the sky in the east is already black while the west still holds the last of the day's colour. She's perhaps 50 years old, which in her world means she has already lived a full life. She knows things that the younger people in her group do not know, things accumulated over decades of observation and experience and conversation with others who were old before her. And she is thinking, in whatever way thinking happened in a mind like hers, about the child just born, about the child's mother, about the years ahead. Not in words exactly, in the sense of hearing a running verbal monologue, but in the organised, sequential, structured way that language makes possible, a series of connected thoughts, each following from the last, about cause and effect and time and probability. The capacity to think in that structured way is itself a product of language, or perhaps more precisely, it's a product of the same neural architecture that language both requires and reinforces. Thought and language are not the same thing, but in the human brain they are deeply intertwined, each shaping the other, each made more powerful by the presence of the other. She takes a slow breath of the cool highland air. Below her, the fire brightens as someone adds wood to it. The new voice of the infant daughter reaches her across the distance, thin and clear. She turns and walks back toward the light. Roughly 50,000 years ago, something accelerated. The archaeological record shows it clearly enough, even if the reasons behind it remain debated. The pace of change in human material culture, already faster than anything in the preceding 2 million years of hominin history, shifted into a higher register. New tool types appeared and spread across continents within centuries rather than millennia. Representational art began appearing in caves across Europe and in rock shelters across Australia and Southeast Asia. Evidence of long-distance trade networks, the movement of specific raw materials, shells, obsidian, particular pigments, across distances of hundreds of kilometres, became more systematic and more extensive. The range of materials being worked expanded. Bone, antler, ivory and shell were shaped into tools, ornaments and objects whose function was clearly not purely utilitarian. This period is often called the Great Leap Forward in older literature, though the term has fallen somewhat out of favour because it implies a single event rather than a process, and because the African evidence makes clear that many of the supposedly revolutionary behaviours of the Upper Paleolithic were present in Africa much earlier, appearing and disappearing in the archaeological record in a pattern that suggests not a sudden revolution, but a gradual, uneven accumulation of capabilities that became more stable and more widespread over time. The argument is not whether modern human cognition appeared suddenly or gradually. The argument is about what drove the change and when the biological preconditions for it were fully in place. One influential view holds that the acceleration visible around 50,000 years ago reflects not a new genetic mutation, but the crossing of a threshold in social network connectivity. As human populations grew and spread, the number of individuals connected through trade and exchange relationships increased. More individuals in a network means more ideas being generated, more innovations being proposed, more combinations of existing techniques being tried. Above a certain threshold of network size, innovation becomes self-sustaining. Each new technique creates the conditions for the next, and the cumulative pace of change accelerates without any new genetic change being required. Language is the infrastructure that makes those networks possible. Without the ability to communicate complex information across social boundaries, to explain a new technique to someone from a different group, to negotiate the terms of an exchange, to establish the mutual obligations that make long-distance relationships sustainable, the network cannot function. A man is sitting in the opening of a rock shelter in the Dordogne region of what will one day be France, approximately 32,000 years ago. It is late afternoon, and the light coming through the trees below the shelter is the particular golden light of early autumn in a temperate forest. Behind him, deeper in the shelter, 3 other people are working, 2 women and an older man. The man at the entrance is watching the valley. He has been here for 3 days, which is unusual. His own group lives 4 days' walk to the northeast. He came here for a reason, carrying with him a small bundle of materials, pieces of flint from a source near his home territory, a handful of perforated animal teeth, 2 pieces of red ochre. He is here to exchange and to talk. The people in this shelter have access to materials he does not have in his own territory, a particular kind of fine-grained stone that can be knapped into blades of exceptional sharpness, and knowledge of a technique for hafting those blades onto bone handles that his own group has not yet mastered. The exchange will not take place through silence and gesture. It will take place through language, through extended conversation, through the kind of negotiation that requires the ability to describe things that are not present, to make conditional statements about future actions, to establish and confirm mutual obligations. He will explain where the flint comes from and why it is valuable. He will demonstrate the blades he has brought and communicate their quality. He will ask about the hafting technique and try to understand the explanation he has given well enough to reproduce it when he returns to his own group. All of this requires the full apparatus of modern human language: displacement, conditionality, reference to future states, the ability to encode and decode complex propositions. This is language doing the work that language does best: moving information across social and geographical boundaries, creating connections between groups that would otherwise be isolated, and allowing the cumulative pooling of knowledge across a community that is larger than any individual's immediate social circle. The man at the entrance of the Dordogne shelter is part of a network. He does not think of it as a network. He thinks of it as relationships with specific people in specific places. But functionally, the relationships he maintains and the exchanges they support are exactly what a network is, and the network is made of language. The cave paintings of this region and period are among the most discussed artifacts in the entire archaeological record, and they are relevant here in ways that go beyond their obvious aesthetic qualities. The paintings in the Chauvet cave in the Ardèche region of France, which are among the oldest well-dated examples of representational cave art, produced by people living approximately 36,000 years ago, show a mastery of visual representation that is genuinely astonishing. The artists understood perspective. They used the contours of the cave walls to give three-dimensional form to the animals they depicted. They showed animals in motion, with multiple legs overlapping to suggest movement. They depicted rhinoceroses, cave lions, mammoths, horses, and aurochs with anatomical precision that implies not just artistic talent, but extended, careful observation of living animals, combined with a cognitive ability to hold that visual memory and translate it into marks on a surface. The production of that art was not a solitary act, or not always. The evidence of multiple hands on the same panel, the presence of scaffolding marks suggesting that painters worked at heights that required constructed support, the careful preparation of pigments using specific recipes that have been partially reconstructed by analysing paint residues, all of this implies a collaborative endeavour that was organised, planned and communicated about. People discussed what to paint and where. They prepared materials together. They evaluated results. They taught technique to younger artists. None of this required the kind of abstract philosophical language that modern humans use when discussing art theory. But it required the ability to communicate about visual qualities, about spatial relationships, about aesthetic judgments, about the intentions behind the work. It required language with enough flexibility and precision to bridge the gap between one person's internal visual experience and another person's understanding of it. The older man inside the shelter has been working for most of the afternoon on a piece of antler, using a small sharp flint burin to incise a series of parallel lines along its length. The work requires sustained concentration and a very steady hand. He produces the lines with the precision of long practice, spacing them evenly, curving them slightly around the cylinder of the antler. The woman nearest to him watches for a while without speaking. Then she produces a sound, a brief vocalization with a rising intonation that functions as a question or a comment. He responds, not looking up from his work, with a short sentence. She laughs, a single low exhalation, and returns to what she was doing. That exchange, which lasted perhaps 5 seconds and will leave no trace in the archaeological record, was fully modern in every relevant sense. A comment, a response, a moment of shared amusement. The social lubrication of a small group spending a long afternoon in close proximity. Language as the continuous background texture of human social life. Not just the vehicle for important information, but the medium in which relationships are maintained, tested, calibrated, and renewed. The man at the entrance of the shelter knows that the daylight is nearly gone. He turns and comes inside. The group rearranges itself around the small fire near the back of the shelter, and the conversation that follows, quiet and unhurried in the gathering dark, covers ground that would have been entirely inaccessible to any hominin living 500,000 years earlier. Plans for tomorrow, memories of last winter, observations about the behavior of the aurochs in the valley, questions about people who are not present. The full range of human temporal reference, past, present and future, brought to bear on the practical and social concerns of a small group of people at the end of an autumn day. The trajectory from the first FOXP2 variant spreading through an archaic human population on the African savannah to the conversation in that Dordogne shelter is long and not straight. Many other genetic changes occurred along the way. Many other anatomical and neural changes accumulated. The social and cultural changes that language enabled themselves created new selection pressures, favouring cognitive abilities that earlier hominins had never needed. The process was circular and self-reinforcing. Language changed behaviour, behaviour changed the environment of selection, changed selection produced new genetic variants, new variants improved language. The mutation in FOXP2 was not a single cause. It was an early and significant contribution to a process of cumulative change that was, in the end, what produced the people in that shelter, with their art and their trade networks and their long-distance relationships and their quiet evening conversation. There is also the matter of what happened when modern humans carrying this full linguistic capacity encountered other human lineages that had somewhat different versions of the same capacity. The contact between anatomically modern humans and Neanderthals in Europe, which appears to have begun somewhere around 55,000 to 45,000 years ago and continued for perhaps 5,000 to 10,000 years in some regions, is one of the most discussed and least understood events in human prehistory. The genetic evidence is clear that interbreeding occurred. All living people with ancestry outside sub-Saharan Africa carry between 1 and 4% Neanderthal DNA. The behavioural and demographic evidence is less clear, but the outcome was not. Within a few thousand years of the contact period, Neanderthals were gone, and modern humans occupied their former territories. Whether language played a role in that replacement is speculative, but not implausibly speculative. If modern humans had a communicative capacity that was meaningfully more powerful than that of Neanderthals, even a modest advantage, the effects would compound over time. More effective communication means more effective group coordination, which means more effective hunting and defense. It means more effective transmission of knowledge across generations, so that each generation starts with more accumulated knowledge than the generation before. It means larger and more stable social networks, which provide more resilience against the demographic shocks, the disease outbreaks and the bad winters and the years of scarce game that periodically threaten small populations. None of these advantages requires a dramatic, obvious difference in communicative capacity. A modest, consistent advantage, maintained over hundreds of generations, is more than sufficient to produce the demographic outcome that the record shows. The woman in the Dordogne shelter is the downstream beneficiary of all of it. The mutation that changed the FOXP2 protein in an archaic hominin on the African savannah 500,000 or more years ago, the slow spread of that variant through the population, the accumulation of additional genetic changes that refined and extended the neural and anatomical infrastructure it initiated, the behavioural changes that those biological changes enabled, the shell beads, the engraved ochre, the long-distance trade, the cave art, the evening conversations about plans and memories and absent friends. She carries all of it inside her, in every cell, in the genome that is the record of everything that happened before her. She does not know any of this. She's thinking about tomorrow, about the walk back north, about whether the river crossing will be passable if the autumn rains have come early. She adds a small piece of wood to the fire and settles her back against the rock wall of the shelter, pulling a fur more tightly around her shoulders. The conversation continues, low and steady in the dark. The mutation is still working. It is working right now in the brain of every person listening to this. The FOXP2 gene in your genome is active in your basal ganglia, in your cerebellum, in the circuits that allow you to understand the words arriving in your ears as a continuous stream of sound and pass them, in real time, into words and phrases and sentences with meaning. The process is so automatic, so deeply embedded in the architecture of your nervous system, that it requires no conscious effort. You do not decide to understand language, you simply do. The same neural infrastructure that FOXP2 helped to build is running quietly in the background of your consciousness right now, doing its work without announcing itself. This is worth pausing on, not in a philosophical sense, but in a purely factual one. The gene that sits in your genome at a particular location on chromosome 7 the gene whose human-specific variant spread through our ancestral population sometime in the deep past, is the same gene that was present in the archaic hominin on the African savanna who first carried the changed version. The specific variants have been copied faithfully, base by base, through an unbroken chain of cell divisions and reproductive events that stretches back hundreds of thousands of years. Every person in that chain, every generation between that first carrier and you, reproduced and passed the variant on. Not one link broke. The chain is intact, and its endpoint is the brain you're using right now to process these words. The history of FOXP2 in modern human populations does not end with the selective sweep that established the human-specific amino acid variants. The gene has continued to accumulate variation at other positions, variation that does not affect the critical amino acids, but that has produced a range of subtly different versions of the gene across human populations worldwide. Some of those variants have been studied in relation to language-related traits, reading ability, phonological awareness, the speed of speech processing, the age of first word production in infants. The results of those studies are complex and often population-specific, and none of the common FOXP2 variants in modern humans come close to having the dramatic effects of the original human-specific substitutions. The architecture is established. The variation now is fine-tuning at the margins, but the gene is still under selection. The human genome is not a static archive. It is a living document, still being edited by the ongoing process of natural selection, operating on a species of 8 billion individuals spread across every habitat on the planet. The selection pressures acting on language-related genes in modern human populations are different from those that drove the original selective sweep. In a world with writing, with recording technology, with the vast external memory systems that human culture has built over the past 10,000 years, the demands on biological language capacity have changed. The biological machinery is no longer the sole constraint on what can be communicated and transmitted. Culture has built extensions to it, prosthetics, from clay tablets to paper books to digital storage, that allow information to be preserved and transmitted without requiring the biological systems to do all the work. In a city in the ancient Near East, roughly 5,000 years ago, a young man is sitting cross-legged on a reed mat in a room that smells of damp clay and lamp oil. He is a student at an aduba, a scribal school, one of the institutions that the Sumerian city-states of Mesopotamia established to train the specialists who could read and write the cuneiform script. He is holding a small rectangular clay tablet in his left hand and a reed stylus in his right. His teacher, an older man sitting a short distance away, dictates a series of signs, and the student presses the stylus into the clay surface, producing the characteristic wedge-shaped impressions that will harden into a permanent record. He's not writing a poem or a philosophical reflection. He's practicing a list of commodities, quantities of grain, numbers of livestock, measures of oil. The text he is producing is administrative, functional, utterly prosaic. It is also one of the most significant things that human beings had yet learned to do. Writing is an external extension of the language capacity that FOXP2 and its network helped to build. It is not a biological capacity, it is a cultural technology, invented independently in several places across the world, and it requires explicit instruction and sustained practice in a way that spoken language does not. Children acquire spoken language spontaneously from exposure. No child acquires writing spontaneously. Writing must be taught, because it is not something the brain is specifically wired for by evolution. The brain systems that support reading and writing are co-opted from systems that evolve for other purposes. The visual recognition of objects, the fine motor control of the hand, the language processing circuits that were already in place. When the student at the aduba presses his stylus into the clay, he is using a brain that was not designed for this task, but that has enough relevant architecture, enough flexibility, enough capacity for learning complex motor sequences and associating visual patterns with linguistic units, to do it anyway. The student makes an error, a sign pressed at the wrong angle, with the wrong number of wedges. The teacher corrects him without irritation, demonstrating the correct form on a fresh piece of clay. The student tries again. He gets it right this time, and the teacher produces a brief sound of acknowledgement, a single vocalization that carries the specific meaning of "that's correct, keep going." The student continues down the list. What is being transmitted in that room is not just the specific content of the tablet, the grain quantities and livestock numbers. What is being transmitted is the technology of writing itself, the entire system of signs and their meanings and the conventions for their use, a system that took centuries to develop and that cannot be rediscovered from scratch by any individual in a single lifetime. The transmission requires language, the teacher explains. The student asks questions. The error is corrected through verbal instruction. The system is passed on, and once it has been passed on, the student who carries it in his memory is capable of reading and writing texts that were produced before his birth, and of producing texts that will be read after his death. The temporal reach of communication, already dramatically extended by the FOXP2-supported capacity for displaced reference, has now become, in principle, unlimited. The clay tablets produced in that school survive. Tens of thousands of them have been recovered from sites across ancient Mesopotamia, and they contain an extraordinary range of content: administrative records, legal codes, mathematical tables, astronomical observations, medicinal recipes, hymns, epic narratives, lamentations, proverbs, and lists of lists. The list of lists is itself revealing. The Sumerian scribal tradition produced long catalogues of words organized by category: trees and wooden objects, stones and minerals, birds, fish, animals, geographic features, the names of gods and kings and professions. These catalogues were used as teaching tools, but they also represent something cognitively significant: the organizing of the known world into categories, the mapping of language onto reality in a systematic and explicit way. This is the kind of thing that only a species with FOXP2 could do, not because the gene directly encodes the capacity for taxonomic thought, but because the neural infrastructure it contributed to building is the infrastructure on which all of these cognitive elaborations rest. The student finishes the tablet and sets it aside to dry. He picks up a fresh piece of clay from the pile beside him and begins the next exercise. He is perhaps 12 years old, and he will spend the next several years in this room, learning not just to write, but to think in the ways that writing demands, precisely, sequentially, with attention to the exact form of signs and the exact meaning of words, aware that the marks he makes will be read by someone who cannot ask him to clarify. Writing imposes a discipline on language that speech does not require. In speech, ambiguity can be resolved by asking. In writing, the text must carry its meaning alone, without the prosodic cues of tone and emphasis, without the immediate social context of face-to-face communication, without the possibility of immediate clarification. This discipline, practiced over years of scribal training, produced minds that thought about language in a new way, that could manipulate it as an object, analyze its structure, be precise about its categories. The scribal schools of ancient Mesopotamia were, among other things, early laboratories for the study of language as a formal system. Thousands of years before those schools existed, the mutations and their downstream effects were already doing their work in brains that had no concept of writing, no concept of school, no concept of recorded history. A woman in the Levant 14,000 years ago, at the very beginning of the period that would eventually produce agriculture and then cities, was talking to her daughter about where the wild grain grew thickest along the riverbank, and how to read the colour of the seed heads to know when they were ready, and which part of the slope retained moisture longest into the dry season. That conversation has left no trace anywhere. It happened, and it mattered, and then it was gone, carried away in the air, dissolving back into the sounds of the valley. The knowledge it contained survived only if the daughter remembered it and passed it on in turn. This is the fragility of purely oral transmission, a fragility that writing was invented precisely to overcome. But oral transmission worked. It worked for hundreds of thousands of years, preserving and accumulating knowledge across generations in every human population on Earth, long before anyone pressed a wedge into clay. It worked because the FOXP2-supported neural architecture made it work. The circuits for producing and comprehending complex speech, the memory systems for encoding detailed procedural and semantic knowledge in linguistic form, the social drives that motivated people to teach and to learn, to ask questions and give answers, to tell stories and to listen. All of that was in place long before writing. Writing made knowledge more durable and more transportable, but the knowledge was already there, living in the minds of people who talked to each other by firelight and by rivers and in rock shelters and on the open savanna. The student at the Adaba in ancient Mesopotamia is the downstream inheritor of that entire tradition. His scribal training is a new technology applied to a very old biology. The gene that allows him to learn the motor sequences of cuneiform writing, to associate visual patterns with linguistic units, to comprehend and produce the complex administrative language of his city's bureaucracy, is the same gene, in all its essential architecture, that allowed a woman on the African savannah to describe the location of safe routes to a child who had not yet encountered them. The technology changed. The biology did not change in any fundamental way. The building built by Fox P2 and its network is the same building, used now for more elaborate purposes than its original architects. The blind processes of natural selection operating on variation in an ancient gene could possibly have anticipated. The last light fades in the room at the Adubba. The student sets down his stylus and flexes his fingers, which are stiff from the sustained fine motor work of the afternoon. His teacher is already gone. Around him, a dozen other students are also finishing, stacking their tablets carefully, covering them with damp cloth to slow the drying and preserve the possibility of correction. Tomorrow they will return and practice again. The day after tomorrow they will return again. Year after year, until the system is learned well enough to be used, until the technology of writing has been added to the biological capacity for language that was already there, and the combination of the two has made the student into something that his distant ancestors would not have recognized, and yet something that is built, in its deepest architecture, entirely from the same materials they possessed. The fire in the corner of the room has burned down to embers. Outside, the city of Nipper, or Ur, or Eridu, whichever ancient Sumerian city this particular school belongs to, is going about its evening business. The last transactions of the market, the smell of cooking fires, the sounds of children, the low murmur of a civilization made possible at its root by a change in a single gene that spread through a population of archaic hominins on the African savannah before any city existed, before any grain had been domesticated, before any wheel had been turned, in the deep time before memory, in the era when the only record of anything was the body itself, and the sounds that bodies made when they turned to each other in the dark. We do not know every step of the process. The science of ancient human genetics is young, and the specific details of when and how and in which population the critical FOXP2 variants first spread remain subjects of ongoing research and genuine uncertainty. What is not uncertain is the outcome. A species that talks, that writes, that records, that accumulates knowledge across generations and builds upon it, that fills the night with language, that has been doing so for longer than any other record survives to confirm. If you have stayed with this until now, you have been listening for something close to 2 hours. In that time, you have heard about a mutation in a gene, about the archaic hominins who first carried it, about the slow cascade of biological changes it set in motion, about the Neanderthals and Denisovans who carried the same variants, about the shell beads and the cave paintings and the evening conversations and the scribble schools that were all, in their different ways, downstream consequences of that single change. You carry the mutation. It is in every cell of your body. Right now, even as you drift towards sleep, the circuits it helped to build are still running, processing the last words of this documentary as sound resolves into meaning, meaning into memory, memory into the quiet of a brain settling down for the night. Sleep well.